Biology, and welcome to a quick video going over how cells use cell signaling to maintain normal control. Now, before we talk about the different ways that we regulate control, I think it's useful that we go to the second page of your document that I gave you and just talk about some review. Stuff we learned about in our homeostasis and cell unit. So there are three aspects to cells communicating with each other to cause reactions. There is the signal reception, there is the transduction, and then there is the response. If you go back and look in Unit 2's folder, there's a PowerPoint with much more detailed information, which I will show you at the end of this video if you want to review. In step 1, hormones in your body, maybe testosterone, maybe growth hormones, maybe cortisol, maybe estrogen, other types of anabolic steroids can signal mitosis. So cell reception is when a receptor protein, like a lock in the cell membrane, binds to a receptor protein, a hormone protein. We call this complex a ligand because they are physically connecting between each other using positive and negative charges. This connection is temporary. It's not permanent. Your cells aren't going to be dividing endlessly. But if you just went to the gym and you did a bunch of lifting and now you're eating quite a bit of protein and you're sleeping, your body is going to release growth hormones telling your myocytes or your muscle cells that, hey, we got to get bigger. We got to have some hypertrophy occur, i.e. some cell division to make our biceps and quadriceps bigger. So that's step 1. Step 2 is the relay of the information. Secondary messengers and enzymes are activated in a pathway 1 to 2, 2 to 3, using ATP, a phosphate group, to bind and activate them. Each molecule works like an assembly line, activating the next one. So this has to be activated, which activates this, and then it activates this. This will not be activated until 2 and 1 are first activated. These pathways in our cells are very specific to mitosis. These proteins are only being turned on when the cell is getting ready to divide in G1 into S. And then that's going to cause effector proteins, transcription factors to be activated that turn on DNA that will get the cell to divide. This DNA will be read to make a protein, and that protein will cause the cell to undergo a step of mitosis. Once we start that chain reaction of going from G1 to S to G2, the party is started, the cell is going to divide. So let's talk about normal control. Your body does not divide unlimitedly, and it does not divide all of the time. It divides at certain frequencies or in certain environments based on need and necessity, because cell division is expensive and it is prone to error. We have checks and balances, much like your parents, your friends, your siblings, or society might check our behaviors or impulses to make sure we don't do stupid stuff. So one of the ways in which we use control is called density-dependent inhibition. If you put a cell in a petri dish all by itself, it will feel the space around it open and it will divide. If you get a cut, your cells will sense that opening in your epithelial tissue, your skin, and you will then create new cells to cover that cut. If your bone is fractured, that space will be felt by your osteocytes, and cells will divide to repair that bone. So density-dependent inhibition is a negative control mechanism. Cells do not duplicate if the other cells nearby it signal the tissue is fully developed. So this petri dish that is meant to show a tissue stops dividing when all of the cells have all of the space taken up. There's no empty space, so no cells will be able to divide anymore. So this is a negative feedback loop. Cells will divide if there is space, and cells will stop dividing if there is no space. They do not divide unlimitedly. The second thing that cells use during mitosis are checkpoints. These checkpoints occur throughout the cell cycle, and they occur at very specific points to make sure that the cell has everything that it needs before it goes into the next stage. Think of this like the checkpoints in an airport. There are 3 checkpoints of note in the cell cycle. The first checkpoint occurs between G1 and S. This checkpoint is all about making sure that the cell is actually pretty darn sure that RGR is occurring. We either need to replace, repair, or grow, hence the cell needs to divide. There is energy available for the division to occur. There's space available for the cell to divide. Everything is good to go for cell division. We then go into S phase, where we duplicate DNA. We make our extracentrioles, so on and so forth. We then enter G2. G2 is the second checkpoint. We want to double check that all of our centrioles have been duplicated, all of the DNA was copied correctly, there are no errors, there are no mutations, there are no problems before we proceed into metaphase. M stands for mitosis, not metaphase. We then enter metaphase, or mitosis, I'm screwing up a little bit. We go through prophase, prometaphase, anaphase, and then we get to mitosis, and the division occurs, and we make sure that we do it before cell division commences and before metaphase occurs. There's 4 different cyclins that I want you to be examining and thinking about what they do to the cell given when they peak, when they start, and when they finish. So cyclin A starts at the end of G2. Cyclin E starts in G1 and it peaks right at the end of G1. Cyclin D peaks in S phase, and cyclin B peaks during mitosis. I hope this helps, and I will see you guys tomorrow.